So let me welcome everybody. Good morning to our grand rounds. Today we have really a fascinating topic and a fascinating speaker to address us on research that's been ongoing in his laboratory for several years, but in the past, I would say, couple of years has kind of emerged in the United States and also worldwide as an area or one of the most exciting areas of research, and one that's attracted a great deal of public interest just because of its history and also because of the nature of this class of substances, and that is psychedelic drugs. Our speaker to sort of talk about that is Dr. L Carhart-Harris, who's at Imperial College London, where he's on the faculty, but just informed us before that he's accepted a position to move to the University of California, San Francisco in the United States in the Department of Psychiatry, and I assume that's Matt State's doing. So good move, Matt, and we'll be doing so soon. But Robin has been somebody who's led the area of research in this ancient class of substances that came to original attention of biomedical research in the medical community, scientific community in the 1950s with the emergence of LSD, the first synthesized synthetic psychedelic that Sandoz produced. And along with that came attention to naturally occurring psychedelic substances like mescaline, psilocybin, and ayahuasca and so forth that were used by indigenous peoples. And the research that was done initially in the 50s and the 60s began a process of exploring the pharmacology and therapeutic applications of the class of substances, but then got derailed when popular culture embraced them recreationally, and Timothy Leary was kind of the high priest, and the cultural political dynamics led to their prohibition for about 4 decades before they began to be restudied again. And they have come to that state in a way which is different than usual new breakthroughs or new initiatives in scientific research do, because it was really not at the behest of conventional academic laboratories or pharmaceutical companies. It was through the efforts of the persistent efforts of advocacy groups that kept the idea of further research and exploration alive, and lobbied for their rehabilitation. As this has been embraced by some academics and some companies have sprung up to pursue development, there's been really a very limited number of leading scientific laboratories that have sought to focus on this area of research. And I would say that probably the 2 leading ones that I was aware of were in Europe, one being in Switzerland with Wallenweider. What's Wallenweider's first name? Do you remember, Robin?
Franz
Wallenweider. And the other was David Nutt and Robin Carhart-Harris, who have pursued the neuroscience of psychedelic drugs as well as their therapeutic applications. I'm not going to go through Robin's CV in terms of history and awards and so forth and so on to save time, but suffice to say that whereas this hasn't been a traditional area of research for the biomedical research sort of mainstream, Robin and David's work has, I think, been the exemplar of it, and it's sort of reflected in an article that was published in Cell by them, which is entitled "The Psychedelic Psychiatry's Brave New World," and whether it proves to be as good as its advertising or not, we'll have to wait to see, but Robin will tell us a little bit about that and the current state of his research on this in the meantime. So please join me in virtually welcoming Dr. Robin Carhart-Harris to update us on his work on psychedelic drugs and their implications for biomedical research and I guess humankind in general. Robin?
Thank you, Jeffrey. Thank you for those kind words and for this kind invitation. I'm delighted to be with you presenting on my work with psychedelics. And what I will try to do is give us a good bit of time for Q&A at the end. I hope there'll be a lot of questions, and I very much welcome that. And I've tried to somewhat reduce this talk down to some of the key themes. It's quite thematic in a sense, somewhat theoretical, because there's some key points I really want to get across. It's a multi-level talk. It starts at the molecular level. I'll go on to talk about brain circuitry, brain networks, and the acute effects of psychedelics as we've looked at them through the lens of functional brain imaging. And I'll also update you about some recent clinical trial research that we've done at Imperial in depression. So starting at the molecular level, and I'm glad Jeffrey covered some of the history because it's so rich, but I think were I to do that, we could easily get lost down that rabbit hole because it really is quite something. So I'm instead choosing to dive straight into the science. So I hope you understand that. So a key starting point when addressing the topic of psychedelics is their pharmacology. And I think through their pharmacology, we can work towards quite a concrete definition of what we mean by particularly the classic psychedelics. And these are drugs that all have some agonist activity at the serotonin 2A receptor subtype. We have a nice PET image here from Gita Knudsen's team in Copenhagen, labeling the serotonin 2A receptor now with quite a selective agonist radioligand. So we're looking at these receptors in their functionally available state. So it's a more informative, more functionally informative image than we've had in the past. And of course, it's in the human brain. So this is pretty much, where it's at now in terms of that general question, where are serotonin 2A receptors in the brain? You'll notice that they're very much cortical receptors, but the distribution isn't entirely homogeneous. You might be able to notice that the expression is relatively lower in unimodal cortex, the sensory motor cortex, and to some extent in the visual cortex. But the expression is highest in so-called transmodal, high-level association cortex. And this is important. We're going to come back to this, but just let that property stick. We know of the importance of the 2A receptor, the critical importance of the 2A receptor to the action of psychedelics, really from work that began in the mid-80s with the discovery by Richard Glennon of the relationship between the affinity, the binding potential, the stickiness, if you want, of a given psychedelic drug, a 2A agonist psychedelic, for the 2A receptor and its potency. So both animal work with behavioral indices of the action of psychedelics, head shakes, or in humans, the effective dosage. And it was discovered that there is this pretty tight relationship between the affinity and the potency of the drug. So to help illustrate that, LSD, very high affinity for the serotonin 2A receptor, very potent, psychoactive in doses down to 10, 15 micrograms. That's a typical microdose for those of you who've heard that term, around about that dosage range. And you compare that with mescaline, which is much more closer to sort of 0.5 gram, 300 milligrams of of that substance and a much lower affinity for the 2A receptor. So that tight relationship between affinity for 2A, agonist properties, so signaling at the receptor, correlating with the drug's potency. And it was Franz Vollenweider who did the first human blocking work, so pre-treating with a serotonin 2A receptor antagonist or blocker, then giving a psychedelic, in the first case psilocybin, but now replicated with a number of different psychedelics, LSD, also ayahuasca containing DMT, another 2A agonist psychedelic, and finding that the effects are markedly reduced, effectively abolished with 2A antagonist pre-treatment. So more compelling evidence for the critical importance of this receptor for the action of psychedelics. And now we have this nice PET work again from the Copenhagen team, plotting the relationship between occupancy of available 2A receptors here with different doses of psilocybin displacing the hot ligand, which is this tracer SYMB36, and a relationship there, as you would predict, of course, with the subjective intensity. But to see it plotted, it's a small sample size, too early to be thinking about non-linearities perhaps, but useful still to know that around about the 60% occupancy range, which is what you're achieving with the dosages that are close to what we use in the therapeutic work, 20, 25 milligrams of psilocybin, achieving that 60% occupancy, broadly speaking, and then that producing these quite intense subjective effects and into the domain of the intensity that's seen in the therapeutic work with psilocybin that I'll tell you about shortly. So that started us off on a solid footing in terms of the critical importance of the 2A receptor. So a natural next question would be, what does the 2A receptor do, somewhat aside from psychedelics? What do we know about this receptor? Well, increasingly, we know that it's involved in plasticity, broadly defined. Yes, we can look at markers of neuroplasticity, and people have, looking at things like BDNF, and a doubling of BDNF in the cortex specifically was a finding from the late 90s. And more recently, looking at different components of synaptic growth, synaptogenesis, spine density, with different psychedelic compounds published in Cell. So increasingly strong evidence there for increases in neuroplasticity with exposure to psychedelics. But then we can look at other functional markers of plasticity. We can look at brain development and a role for the 2A receptor there. Very exciting finding I'll tell you about in a moment, linking 2A signaling to the proliferation of basal progenitor cells, and that being linked to the evolution, to brain evolution itself, and a role for the 2A receptor there, recently published in Neuron. We can also look at learning, low-level learning in particular, and yes, the classic U-shaped dose-dependent relationship here, but a certain dosage has been shown to enhance low-level associative learning, aversive learning with air puffs in rabbits in particular, but also reversal learning, extinction learning, extinction learning being increased with exposure to psilocybin, actually. We could also look at reversal learning and cognitive flexibility and post-acute, subacute increases in reversal learning after ayahuasca. Recent findings there. The acute data is a little bit mixed. I think there's probably a dose dependency there. Some work showing decreases in cognitive flexibility, and also we have some work showing increases, but I think the relationship post-acutely is more compelling, and we have data demonstrating that as well. 1 month after psilocybin, increases in cognitive flexibility. Moving up a level of abstraction again to psychological flexibility, that construct that is popular and has been popularized really by the acceptance and commitment therapy model, psychotherapy model. So the ability to accept difficult emotions and memories, sit with them rather than seek avoidance strategies, has been shown to be increased after exposure to psychedelics, increases in psychological flexibility. Here's just a chart from that rabbit work showing accelerated learning rates with exposure to LSD, and that being slowed down with ritanserin that has some 2A blocking properties. So this is the recent paper on 2A signaling increasing the proliferation of these basal progenitor cells. Whether we can translate that into the adult human brain is, of course, another question, but that it was found in embryonic cortex is still very interesting and fits in with this notion of signaling at the 2A receptor having these plasticity related properties. And so if we further put psychedelics to one side and drill down a little deeper into 2A receptor and functions associated with it, there's some intriguing evidence that chronic stress upregulates the 2A receptor. It sensitizes it to signaling. It increases its availability, but also its sensitivity to signaling with different readouts of that. So we could think of chronic stress as priming this system, the 2A system. And then the most reliable way that I'm aware of to trigger the release of serotonin is through acute stress. And so here we're looking at tail pinch handling, swim stress, a range of different stressors, and marked release of serotonin onto that primed system. The natural thing to assume would be a subsequent increase in plasticity. So what I'm proposing here is, in a sense, this is why the 2A system has evolved, why it's developed, what its function is, that it is a system that engages for markedly enhanced plasticity under adverse conditions when adaptability may be critically important. And I think this may well be a fundamental about this system that is being hijacked, in a sort of unnatural way by psychedelic drugs, and helps us understand and contextualize, I think, the action of psychedelics. And based on that, I recently proposed this model I call pivotal mental states that ties in with these properties. And here it's a model that says that the hyperplastic, these hyperplastic states induced by serotonin 2A receptor signaling can be thought of as hypersensitive to context, hypersusceptible, I suppose, to environmental conditions, whether from the outside world is the most obvious place to look, but also within in terms of memory, emotion. And so you have this sensitized agent, if you want, that is going to be exceptionally sensitive to the conditions that it finds itself in. And if those are favorable, enriched, if you want, like a therapeutic context with compassionate support, perhaps priming, positive priming ahead of the experience, positive expectancy, then it may well be logical that hyperplastic agent in those conditions would pivot towards improved mental health outcomes. And indeed, that's what we're seeing with psychedelics in population data and controlled research. But imagine those perhaps anomalous cases where someone may have received a psychedelic drug without any awareness. Maybe their drink's been spiked, and perhaps they're in an unfamiliar environment with unfamiliar people, certainly not a therapeutic context, and they're in that hyperplastic state, and things take a pivot, in a negative direction. So this is the model. It's very simple, I know, but I think it could be a very fundamental model of how we think of the action of psychedelics, that there's a danger that this model could be overlooked, and we think that the drugs themselves have intrinsic properties, and we don't need to consider the contextual factors, and that might not be correct. Time will tell. So here's an overview of the situation with the latest human research and clinical trial research into psychedelics. And I just want to emphasize a couple of key take-home messages rather than get into the weeds of these different studies. And the first one to mention is that all of these trials, all of these studies, including some healthy volunteer research, have manipulated the context in a positive way. I think across the board, they've all had music listening. That's a detail that could easily be lost here. I think the patients in most of these studies and trials have had two people attend to them, supervising them throughout the whole session, sometimes also for at least 2 visits to prepare for that dosing session itself, and also often 2 or 3 or more visits afterwards to talk through the experience. So they get a lot of attentive listening, a lot of care, and the sessions themselves are very long, and so it'd be easy to overlook these contextual elements and just look at some exciting findings in terms of reliable drops in depression scores, for example. So that's the first point to emphasize. The next one is that indeed the changes, the improvements that we've seen in our depression research and others have seen looking at end-of-life anxiety and depression and also addiction, the improvements are generally rapid. We see them the next day if you revise your measures to work in that way, and at 1 week. And they also appear to be sustained. And I mentioned that really in relation to other treatment models, whether they're the classic chronic medication treatment models, or even ketamine therapy, where there is repeat administration, typically 6 sessions in a condensed period of sort of 2 to 4 weeks, and then often further treatments. And so this is quite different. It's often just 1 administration session, sometimes 2. It was 2 in our most recent depression trial that I'll tell you about at the end of this talk. And so this is quite a unusual finding and model that outcomes could be improved in a sustained way like this, well after the drug has been washed out of the body. What accounts for a potentially enduring change like that is a fascinating question. So also a range of different disorders would probably be the last take-home point here. Small trials, they are very small. They're just pilot studies, a lot of them. They're not always placebo-controlled. Our TRD trial wasn't, it was open label. So a lot of design limitations and small samples, but a lot of different indications are being looked at. And as I'll tell you about in a moment, I think there may be a model that can be proposed to potentially make sense of this so-called transdiagnostic action of psychedelics. And it's this, and so the model is that in a range of different mental illnesses, diagnostic categories, you will see the symptoms being characterized by a kind of routing or canalization of cognition and or behavior whether it's repeated behaviors in OCD, or repeating thoughts, ways of looking at oneself in eating and image disorders, excessive control, repeated behaviors in addiction, and the specificity of whatever that object of relief is. And then we can think of specific anxieties, and how there is, often one specific notion or theme that is that trigger of the anxiety. We can think of ways of looking at oneself with chronic pain and other functional neurological disorders, and also the negative cognitive bias in depression. And so the model here is that there is a common space, and it is this entrenchment of ways of thinking and behaving that are targeted by psychedelic therapy. First of all, that's how I'm proposing these disorders are characterized and what the common ground is, but then that the intervention with psychedelic therapy comes in to relax these overweighted habits of mind and or behavior. And that represents a window of opportunity. It's not intrinsically therapeutic, but it's just an opportunity to work on the healthy revision of those habits of mind and behavior. You could think of it as a kind of, very broadly speaking, shaking up the system, and then that being an opportunity that things settle, things recalibrate into a healthier form after that acute challenge, if you want. So here I want to now turn our attention to our brain imaging work with psychedelics, and a series of slides that are essentially looking at the same property. Here with psilocybin, looking at between system communication, and that being increased under acute dose of psilocybin in healthy volunteers. Resting state scan, so in this case I think eyes open, resting state. And we've ran these analyses a number of different ways here: seed-based resting state functional connectivity analysis, seeding V1 via retinotopic mapping, and then asking, where in the brain is activity correlated in its spontaneous nature with V1, and seeing that, as you would expect, and as is well known, that activity is generally coherent with V1 within the rest of the visual system, but under LSD there's much more of the brain contributing, if you want, or correlating in its activity with activity in V1. And this effect correlated with the subjective experience, and specifically ratings of complex closed eye imagery. We've also looked at, again, running the analysis in a slightly different way, looking at global brain functional connectivity or functional connectivity density, asking where in the brain does activity become any less or more correlated with activity in the entirety of the brain under each condition. And here under DMT, we see that these regions colored in yellow are increasing their coupling strength with the rest of the brain under the DMT. Now, it'd be easy to see a finding like this and infer that this system patch, or this network, it's a very broad frontoparietal pattern, encompassing actually probably a number of different frontoparietal networks. But it'd be easy to look at that pattern and imagine that it becomes more prominent under the psychedelic, and actually that's somewhat misleading. And so here, using a leading eigenvariate dynamic analysis in collaboration with Morten Kringelbach at Oxford and Gustavo Deco, we've looked at and Joanna Cabral, we've looked at the visitation of these different brain states under under DMT as we have with other psychedelics. And the findings are quite consistent that a globally coherent state, somewhat misleading in the fact that there's no color in this brain. In a sense, we should see the whole of the brain colored there, because this is a globally coherent brain state. So the visiting the predominance of this state increases under the DMT. This globally interconnected state increases, and that is consistent with those images I was showing a moment ago. You know, the classic one with the circles and increased communication across these different systems. That's very consistent. But when I flagged that functional connectivity density result, this one, as being somewhat counterintuitive, that you think this network is sort of dominating the picture, it's actually the opposite. It's sort of being skipped over under the psychedelic. And I think that's very interesting. You could say skipped over, or rather its form is more sort of melding in with the rest of the brain. We're losing this and it's becoming more of a globally coherent state. So in a sense, it's segregation or separateness or modularity from the rest of the brain decreases, becomes more smudged in with the rest of the brain under the psychedelic. And that's a critical nuance, I think, when thinking about the functional brain effects of psychedelics. Now, historically, I think partly it's been my fault and others, we've been somewhat swept up with this relationship, really because of how, you know, some of the literature on the default mode network and its relationship to high-level functions, self-reflection, imagination, and such like. It's been very easy to intuit that it would be affected by psychedelics. Indeed, it is. That's not what I'm saying here. It is. It's just, I think the narrative has been somewhat too default mode centric on the action of psychedelics. As you saw, these patterns that are being skipped over, yes, the default mode, but also these other frontoparietal networks. And this increase in functional connectivity density is not just the DMN, it's other frontoparietal networks. So that's the detail that I just want to emphasize. Yes, we have seen relationships between a decrease in the integrity of the default mode network here, actually a specific posterior circuit within the DMN correlating with ego dissolution. We've seen other things like increases in functional connectivity density in our LSD data. So an increase in global brain connectivity correlating with the intensity of the experience and specifically ego dissolution. But I just want to caution that perhaps the relationship isn't selective to the DMN. It's more sort of DMN plus, you might say. And that's a preamble to this. Let me just turn that off if I can, which is to look at a number of different properties of the human brain, whether it's global brain function or anatomical features like myelination. And so let's just run through some of these. First of all, there's a reminder of where the 2A receptor is in the brain, in the human brain, and that very strong expression in the high-level transmodal cortex. And now if we look at, and now there are a number of different maps, but typically the data comes from macaque to human comparisons, and then mapping where the expansion has been greatest in that evolutionary chain from macaque, where we have the most data, to human. And we've formally plotted the relationship and found that indeed it is, as you would intuit, just eyeballing these images, that there is a tight relationship between where the 2A receptors are, remember, key receptors for the action of psychedelics, and the expansion of the human brain. And then I told you about that recent neuron paper looking at proliferation of basal progenitor cells. So I think, you know, there's something here that serotonin signaling at the 2A receptor, perhaps under conditions of environmental stress, has you know, contributed to our you know, unique phenotypes and our unique adaptability. It's a fascinating model, and in a sense, that's the point of this slide. Everything else is just sort of dressing for that central theme. We can look at the principal gradient in the brain that separates so neatly the unimodal cortex from transmodal cortex. We can look at connection length and the longer fibers in this pattern, this frontoparietal pattern. I playfully call it the human system. I hope you'll allow me that. We can look at intersubject variability and functional connectivity and see that that variability between individuals is higher in this higher level system. We can look at lighter myelination in the system. We can see that the system is more concerned with processing abstract features as opposed to concrete, that it processes longer temporal windows, and that it is markedly affected by the action of psychedelics acutely. So a lot of food for thought there. You know, I think time's needed to kind of crystallize the details on it, but there's something very interesting here, linking 2A signaling to cortical expansion, and a way to understand, you know, why and for what has that happened. So the remainder of my talk really is about a model of the therapeutic action of psychedelics, and also to tell you about our recent trial that's now completed, written up and accepted, and we anticipate publication perhaps in April. We don't have the date yet, but so I want to tell you about that trial, and usually I stop short of mentioning any findings because it wouldn't be right to do so with the paper still being processed. It is accepted now, so I'll tell you about one or two findings. I won't show any figures, but I'll tell you about some critical findings there. So the model, before we get into that, is one that is inspired by hierarchical predictive coding or processing, a model of mind and brain function popularized by the likes of Carl Friston. Of course, many people have been inspired by this model, but Carl has personally inspired me much in thinking about the brain in this way. And it has been Carl's work really has been the main inspiration for applying this model to an understanding of the action of psychedelics. So a few little kind of cute examples of hierarchical processing in the perceptual domain. Here we can read texts like this, despite the details, the letters themselves being somewhat jumbled up. The first and the last are actually in the right order, and because of that, we can group, we can form the gestalts and the wholes and read the text. So it's an example of the way our minds and brains take shortcuts for reasons of obvious efficiency. And the point is that that is likely instantiated in a hierarchical manner. And optical illusions, things like this, where if you were to see something like this in the world, in the universe, then there would typically be motion. There isn't any, it's static, and yet we hallucinate the motion because there's something about the image that tickles our internal models that tell us we should be seeing motion. We're not, but that's what we hallucinate. And so it's a kind of cute example of that predictive processing in the brain. To call it hierarchical is a sort of, you know, next inference, but there's various evidences that support that, and intuitively, of course, it's long made a lot of sense. So one way that we can look at how hierarchical information processing is encoded in the brain is to look at traveling waves. So this is a recent development in terms of modeling traveling waves in this way that links it to predictive processing. And the simple example to give of the principle is to look at visual stimulation, as simple as closing one's eyes and opening them. And as we close our eyes, the direction, the predominant direction of these traveling waves, like, you know, ripples in a still water after dropping in a pebble, they have a direction, they move from the center out. So it's a similar kind of principle here, looking across the cortex with EEG. And here the hierarchy would be high end at the frontal cortex to the lower end with the sensors in the over the occipital cortex. And so with eyes closed, the dominant direction of flow of these traveling waves is from the front to the back, top down if you want. And then one opens one's eyes and the direction flips. It goes bottom up. Why? Because sensory information's coming in and then it's traveling up the hierarchy as it's being processed. So that's the switch. It's well demonstrated and also modeled. And so when I discovered this work, and actually in particular Carl Fristen's commentary on it, which linked it to hierarchical predictive processing, really commented on the modeling work related to that, I reached out to the authors and said, oh, we've got some great psychedelic drug electrophysiology data. Would you be interested in analyzing it? And we have a strong hypothesis that the direction of the traveling waves would be flipped from top down to bottom up. And the data that we passed over was our DMT data, where there is this fascinating quality to the subjective experience, to the phenomenology, where people describe the appearance of very, very vivid and immersive and often complex and well-formed visual information with eyes closed. They report things like encountering seemingly sentient other beings, alien-like beings, or semi-human beings, very strange things, but very, pretty common. About in the dose that we typically use in our DMT research at Imperial, just over 20 milligrams of DMT, it's about half of volunteers will report these vivid entity encounter experiences. And so that was our hypothesis, that was our model coming in, that we would see a flip in the direction of the traveling waves as you see when you simply open your eyes. And that was very much supported by the data, remarkably so, and also with a very tight relationship with the subjective ratings as well. There are other ways to scrutinize hierarchical properties in brain function. We can look at gradients in the brain, the separation of the unimodal cortex from the transmodal cortex when looking at functional connectivity. We've done that, and there we've seen what we had hypothesized again, which is a flattening of that hierarchy. And how should that translate phenomenologically? Well, I think it can make sense of a lot of the phenomenology, whether it's the relaxation of the internal models themselves, the relaxation of the assuredness with which we experience those models, the confidence that we have in our models of the world, of things. And ego dissolution, I think, is a good example of that assuredness, that self-assuredness that we typically have, that we're a particular way, a particular person, others reinforce that narrative, that gets stamped in as we develop. That's something that relaxes in this, profound way under psychedelics. It's overlaps much with the phenomenology of meditative experiences and also pathological experiences linked to psychosis, for example, particularly early psychosis, the dissolution of self and expansion and sort of fraying of ego boundaries dissolving, and then expansion of self subject with other objects. And so, yes, it makes sense of that high level construct, I think. And then we can look at other features like how memories and emotion, and as I described, these strange, very strange features like entity encounters can manifest into conscious awareness. And the model would be that these are memories, images of the mind that are usually held down. They're usually suppressed. They often have an archetypal quality. These images that percolate up, and so the model would be that these images are usually held down. And if one was faithful to the language of predictive coding, you would call them prediction error. I've often struggled with the nature of that term. I prefer to call it just signal or bottom-up signal. I know it's not entirely faithful to the model, but I just think it's too counterintuitive to call some of these experiences, errors and prediction error. It's more information that is allowed to percolate up that usually is explained away and suppressed. But anyway, that's the model, and it's encapsulated by this REBUS acronym that is phonetically correct, relaxed beliefs under psychedelics, if not entirely faithful to the first letters themselves. So there we have it, and I promised I would tell you about the trial, and I think we're okay timing wise. And it was a 2-arm trial, double-blind, randomized control trial in 59 patients. Yes, we wanted to do 60. Circumstances thwarted that. So 30 psilocybin, 29 escitalopram. The design was this, that there were 2 arms. And so if you went into the psilocybin arm, you would receive 2 25 milligrams of psilocybin 3 weeks apart. And the very next day after your first dosing session with the 25 milligrams of psilocybin, you had a pot of capsules, and these capsules were placebo capsules, microcellulose, inert placebo. You would take 1 for the first 3 weeks and 2 for the final 3 weeks. So placebo, daily placebo capsules for 6 weeks. There was psychological preparation, supervision of the sessions, and at least two integration sessions after these dosing sessions with psilocybin. If you went into what we call the escitalopram arm, you did actually receive psilocybin, but you received a very low dose, and that was intentional, that we give some psilocybin because of the strong expectancy that we hypothesized with the psilocybin. And so that allowed us to standardize the message to all of our participants that they would receive psilocybin without any deception. You will receive some psilocybin, it's just the dosage might differ. And if you went into that escitalopram group, yes, you did get a dose, but it was 1 milligram, and that is presumed to be inactive. Relatively safe assumption. We've looked at EEG data in healthy volunteers with 1 milligram versus pre-dosing baseline and not found any effects. And in terms of the escitalopram, so the day after your first 1 milligram session, the psychotherapy, the psychological support, was entirely standardized. Sure, its quality might be different, but the quantity was standardized. You would receive escitalopram the very next day, 10 milligrams for 3 weeks, and then 20 milligrams, 2 capsules for the remaining 3 weeks. So we call it psilocybin versus escitalopram, but there's a bit more nuance to it because of how we try to control for the therapeutic component with psilocybin therapy. At this point, I usually just tease people, but now I'm going to give you a sense of the findings. It's just been accepted by New England Journal of Medicine. The response rates according to our primary outcome measure, which was the QIDS, the Quick Inventory of Depressive Symptoms or Symptomatology, self-report at the primary endpoint, which was 6 weeks. So this was a 6-week trial period. Response rates were 70% with psilocybin and just under 50% with escitalopram. That intuitively you might think that that would separate. It didn't actually, didn't quite separate. Remission did. Remission rates, which are scores of 5 or below on the quids, remission rates were 57% with psilocybin at the primary endpoint, and I think 29% with escitalopram. So more or less twice the remission rate with psilocybin and escitalopram, and that separated. Where we planted our flag, as you're of course meant to do with clinical trials, was on the mean change in quids. That didn't separate. So we've been well marshaled, and I can live with it by the journal to report the trial as a negative trial based on that primary outcome measure. The secondary outcomes very strongly favor psilocybin. The tolerability, the side effect profile, depends how you look at it, but is somewhat comparable between the two conditions, but different in nature. So yeah, hopefully you'll be able to read that manuscript in the not too distant future, and scrutinize it as you should, and maybe it will move the needle along in terms of our understanding and our ability to contextualize. I think that's the key thing. Going up against the standard treatment, albeit somewhat of a short course of an SSRI, but still, if they're not working well enough by 6 weeks, yeah, but it helps to contextualize, I think, the promise of psilocybin therapy, still with limitations, somewhat selective sample, because most people coming in really wanted the psilocybin therapy, but very difficult to control for all of those factors. But yeah, so I'll draw it to a close there. Thank Jeffrey again for inviting me to deliver this Grand Rounds, and for your attention, and for the broader team that have made this research possible and have carried it out, and also those who backed our center at Imperial. Thank you again.
Thank you so much, Robin. That was a really terrific talk, and such an exciting area of research. And congratulations on the acceptance of your paper. We really look forward to reading it. I had a question, but I actually wanted to first turn it over to Dr. Lieberman, who I think may have to leave fairly soon, but I don't know.
Yeah,
Dr.
thanks.
Lieberman, if you have a question you want to get in before heading out.
Yeah, thanks, Jonathan. I'll ask a quick question about the paper. So, Robin, you've really got a tiger by the tail with this area of research, because it really offers, this class of substances offers.
Yeah, I think your signal is.
Am I being heard okay?
I can hear Jeffrey.
Is that okay, Jonathan?
Your signal dropped out. Okay. Oh, go ahead. Sorry.
So you've really got a tiger by the tail with this area of research, because these substances offer so many possibilities, both from a neuroscience as well as a therapeutic perspective. But from the therapeutic perspective, and specifically the study in the New England Journal will come out, one question would be, is the use of these relative to standard antidepressant treatments specific to treatment-resistant patients, to any particular symptomatic features or subtypes of depression, or was this sort of a garden variety MDD group in general? And then the second part of it is, were there any adverse reactions of any sort?
Yeah, I'm glad you asked that question, because I don't think I made it clear enough what the sample's characteristics were. It was indeed major depressive disorder, and the criteria for entry was a HAM-D score of 17 plus at screening. That shifted, that reduced when it came to pre-trial baseline, but that would be moderate depression. It was more of the sort of broad heterogeneous type of depression, so not TRD specifically. And the severity was, although there was a range into severe, it was sort of mild to moderate, probably dominated the sample. Some treatment resistance in there if you go with liberal criteria of it, so past history of at least 2 different medications, but still people who had quite light histories of treatment. So very general MDD, major depressive disorder. And then adverse events, there was nothing that stood out as sort of frightening in any way related to the sort of specters, as I call them, that hang over psychedelics, the idea of triggering psychotic symptomatology. Sometimes people think that these are addictive substances. They're not. So people sometimes think, oh, well, you're giving them a drug, they could become addicted to it. There's no evidence of that. There isn't any evidence of that generally. HPPD is another specter, that's hallucinogen persisting perceptual disorder. In most people's vocabulary, that would translate into flashbacks. So the idea that after you've taken the drug, you continue to have aberrant, particularly visual perceptual effects. We didn't see any of that. So nothing that was really sort of frightening. The most prevalent adverse event in the psilocybin condition were headaches, transient headaches. And the prevalence of that's pretty high. It's about 40%, and quite consistently, around 40% in the different studies that have been done. Seems to be dose dependent. Seems to be more common with the higher dosage. It's transient. They're transient. They often subsided by the next day. 2 days post. But beyond that,
any GI effects like nausea or vomiting?
There was some nausea during, so some acute nausea with the psilocybin in a couple of cases, I think.
So I turned my video off. My Wi-Fi seems to be a bit unstable. With the video off, it seems to work better. So apologies, but there are a bunch of questions, so I'm going to start going through those. So the first question comes from Ralph Wharton, who asks about the Hopkins studies by C. Savage and use of LSD with alcoholism, and also inquires about the risk of flashbacks, which I think you did address.
Yes, yeah. I mean, the historical work, as Jeffrey was saying in our pre-meeting, was kind of dominated things, that particularly alcohol dependence. LSD therapy for alcohol dependence was probably where most of the research was in the 60s. So yeah, that's a major domain that is going to be looked at. Really, the depression, I think more than anything else, has been the symptom cluster, if you want, that has dominated people's focus in this sort of more recent era of psychedelic research. But there's a lot of potential, I think, in addictions. Matt Johnson's done some great work there, looking at tobacco addiction. Michael Bogenschutz with alcohol dependence. And there's more to come. I know Peter Hendricks at Alabama is doing some great work looking at cocaine, psilocybin therapy, I think, for cocaine addiction. So it's just that, this all of this work's being processed and written up. But there's some exciting findings to come in the addiction space. And the other component of the question was?
The other aspect of the question was the risk of flashbacks, but I think you did.
Yeah. Well, I could say a little bit more, which is that we, most AE reporting is you keep it very general. Were there any side effects? But we thought it would be probably good practice, given the stigma and the concerns around psychedelics, to drill down a bit with the knowledge that people would ask. So we actually had things like a self-constructed scale that referenced the DSM definition of HPPD, flashbacks, if you want, hallucinogen persisting perceptual disorder, and also manic episodes, just in case, we triggered some hypomania or mania even. so the DSM definition of a manic episode, and saw only decreases in the manic item, and no cases whatsoever of any HPPD, even after specifying it. So no cases in this trial. There's some suggestion from observational research with bigger numbers, into the hundreds, that the prevalence might be higher with LSD of HPPD than psilocybin. You do hear about it anecdotally, so I don't want to dismiss HPPD, but its prevalence seems to be very low, and it might be more prevalent with LSD than psilocybin.
Great. The next question is from Adam Klein, who asks, do you believe that the phenomenology of the psychedelic experience is necessary for some of these therapeutic effects? Or is there an underlying pharmacologic effect that is primarily responsible?
Yes, this is the classic question now, and particularly with David Olson's and colleagues' paper in Nature that addresses this in terms of animal modeling, and suggests that you can get a antidepressant outcome or a positive outcome in terms of therapeutic-like outcome without any of the classic acute behavioral effects that you typically see with these drugs. I just wonder whether that will translate into humans. All I can say is that in the human research, the weight of the evidence is very strongly in support of the experience, the subjective effects, the quality of the experience playing a role. And it's not just intensity, it seems to be something about the quality of the experience and the parameters that are coming through that seem to be important. There's an issue with the wording. Mystical type experience is one of the scales. We have an emotional breakthrough inventory. That seems to be highly predictive, as predictive as the mystical type experience. And then Hopkins have recently developed an insight scale for the acute experience. We're in the process of developing a post-acute insight scale. So these are what I call, they're mediators, they're vehicles through which I think you need to pass in order to get to the desirable therapeutic outcomes. And that's what the weight of the evidence is saying. Now, that doesn't say that it's all psychology and not the brain, because of course those things are tightly coupled. But it might say that it's more than just, molecular pharmacology, or rather we can think of dynamic, dynamics in global brain function that might be a good place to look in terms of a meaningful neural correlate of these subjective experiences. Great.
The next comment is from Dr. John Mann, who writes, fascinating talk. Suicide is associated with more 5-HT2A agonist binding in our studies. What happened to suicidal ideation in your RCT?
Yeah, it was actually, it separated, meaning there was a greater decrease in suicidality with psilocybin than escitalopram. These secondary measures weren't corrected for multiple comparisons, so that's the caveat there. But it did separate in favor of psilocybin. And this isn't a new finding. There's other work, population level and controlled research data suggesting rapid and marked improvements in suicidality via psychedelic therapy. And the population data wasn't psychedelic therapy, it was psychedelic use. Peter Hendricks has something nice on that. And more recently in observational data, so again, bigger samples, hundreds, we saw significant drops in suicidality. Rick Seifman is the first author of that work. So it seems to be a good action on suicidality with psilocybin therapy in particular.
Great. Daniel Umbridge writes, great talk, and then asks a question about your rebus model, pointing out that psilocybin does not seem to have any effects on mismatch negativity, which is a measure of predictive coding, and wonders whether your model might apply specifically to visual modalities.
It might. We published on auditory mismatch negativity with psilocybin, I think it was, MEG work. Chris Timmerman is the first author of that. And there was an effect. It was in the direction of, gosh, I think reduced surprise signal and a greater signal to the tones. What was it? But it was a diminished surprise signal. And there the model is that you entrain the prior with the standard, boop, 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 boop. So that's developing your prior, and then beep. That's where you should get the surprise. And so if you're relaxing your prior, if you're not stamping in the boop, 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 then you would argue that you would be less surprised when a surprise comes along. And that was the finding that we had. So we did interpret that as consistent with rebus, but it's open to interpretation. I actually find the traveling waves evidence a bit more compelling. There's other ways to scrutinize these data, and that's being done now. Some collaborators are doing that with controlled experiments in primates, and also translating with some human visual perceptual tasks. But Olivia Carter looked at binocular rivalry and found a greater proportion, I think with Franz Wollenweider, and found a greater proportion of mixed percepts under psilocybin. And again, that would be consistent. So neither prior is being weighted. It's not this one or that one. It's an even playing field. Yeah. So it's certainly right to intuit that we could come in with a bit more experimental control to scrutinize the rebus model. It's just difficult to make experiments. It's difficult to make standard experiments sort of apply as they typically do to normal waking consciousness with psychedelics, through like a generalized deficit confound, like you have in certain mental disorders like schizophrenia and depression.
Right. Susan Bond writes, you spoke about plasticity in the sense of extinction, but has memory reconsolidation rather than simple extinction been studied?
Yes, memory consolidation and extinction has been studied with psilocybin in rodents. Yes. If I caught the question correctly, I hope so. And,
sorry, go ahead.
I'd probably cite it. I'm happy to share my deck, and I probably cite it there, and perhaps I could try and look it up for us so that I can share that.
Okay. Yeah, if you're able to share your deck, that would be terrific, and then I can distribute that to people who are interested.
Yeah, happy to do.
So the next question is from David Hellerstein, who asks if there is any data on the effects of psychedelics on habit learning and related circuitry.
Habit learning in rodents. Or animals. I think there is the study that I wanted to remember and have just looked up is Catlow et al. 2013, that involves psilocybin, different dosages, and learning and extinction learning. I think there's more now, and I think there's something with DMT. I wish I had it on the tip of my tongue, but I don't quite. But maybe I'll find that for you as well.
Great. Yeah, no problem. You have a lot of information that you're conveying, so totally understandable. The next question is from Brian Fallon, who asks if you could provide your impressions of MDMA therapy for PTSD?
Yes, well, it's somewhat related. It's similar but different, as is often the case. So MDMA isn't a direct serotonin 2A receptor agonist, but it hits the serotonin receptor, hits the serotonin system hard, markedly increases release of serotonin. Yes, it releases other neurotransmitters, dopamine and noradrenaline, but not to the same extent as serotonin. Really, that's what sort of defines its pharmacology, is that particular effect on the serotonin system and serotonin release. And so, all of the different serotonin receptors and noradrenaline and dopamine are going to be implicated. And I think that bears out in terms of the phenomenology, that it's less of a sort of, in a sense, it's less cognitive, it's less dramatic, it's less intense, but it's more effective. These drugs, MDMA is described as an empathogen, that it increases empathy, and there's growing evidence all the time of its effects on social cognition, social behaviors, pro-social behaviors being increased with MDMA. And so there's an opening of people's emotion and willingness to confront difficult material, difficult memories, past trauma, and share those traumas, talk about them. And so there's more talking in MDMA psychotherapy than there is in classic psychedelic drug therapy like psilocybin therapy. The psilocybin therapy experiences are typically eyes closed, listening to music, minimal dialogue, because people would very much struggle to talk and really wouldn't want to in the throes of the peak of a high dose psilocybin experience. But MDMA seems to invite that more. There is more dialogue, and it's a bit closer to traditional psychotherapy with the dialogue, and that's where some of the difference lies. And I think with the big release of serotonin hitting other receptors like 1A receptors in stress circuitry, an inhibitory receptor, and so when that's being hit postsynaptically in stress circuitry, that's going to, broadly speaking, reduce activity, reduce responsiveness in that stress circuitry. So that might explain some of the increased ability to go there, to go to those difficult places with MDMA. Yeah.
Great. The next question is from Fletcher Bell, who asks, how do you envision psychedelics being employed clinically, especially given that antidepressants such as SSRIs are primarily prescribed by primary care physicians, often in already under-resourced areas?
Yeah, I think we probably will get some clues from how ketamine's being rolled out. I think it will be clinician administered, and it should be. I don't think this is a treatment that is for home use. That things are going to go awry if efforts are made in that direction. In my view, even with microdosing, that's a path that's fraught with complications. If people think you could have something a bit more like a chronic medication dosing regimen, and you can do that at home, and you have the drug with you at home, it's going to be a tough sell. But clinician administered could work, and that's how I see it. And the interesting question becomes, is this just for depression, or are there other benefits in healthy individuals? Could it even be prophylactic? So I think there's a bright future in that space. There's a danger that the system can open up ahead of the evidence, as happened with medicinal cannabis, but we just, in a sense, as a researcher, we need to keep the research close to any opening up. So things like the Oregon Initiative, legal psilocybin therapy and magic mushroom therapy in the state of Oregon, although there's a runway, and that's until that's actually available and it's being overseen by the Oregon Health Authority. But if that's tightly coupled to research and real world data collection, then that's going to strengthen things, and that can work. So yeah, there's a responsibility, I think, for the researchers and the scientists to make sure that they're tightly coupled with any opening up. But to be a bit briefer, clinician administered in clinics, and with training and a protocol, is the way that this, people are envisaging this is going to be developed and rolled out.
The next question is from Holly Huntsberger, who asks whether you think there could be potential use for these drugs with neurodegenerative diseases where depression and anxiety are often comorbid.
Yeah. So potentially it's just learning to walk before we can run. But now that commercial money's coming in, there are people looking for novel angles, and that's one of them. I mean, there is a logic to thinking about potential there. If you're just kind of hedging your bets and saying, yeah, but we're treating the mood comorbidity there, or you're being brave enough to say that maybe even some of the sort of core symptomatology of the neurological disorder could be treated, whether it's through some anti-inflammatory action of the psychedelics. Some early data in that direction exists, although I haven't seen anything yet compelling in the brain, difficult to measure. But there is some indirect evidence I'm aware of reduced inflammation elsewhere in the body with 2A agonist psychedelics. So it's an interesting one. I wouldn't rule it out. Yeah.
Megan Finsas asks, or writes, curious about whether you think 5-HT2A expression in the periphery also might play a role in psychedelic effects. And then thank you for a great talk.
Thanks. Yes, likely. I mean, there's this term that you hear among users, if you want, of body load. And some of the more selective 2A agonist psychedelics, like these N-bomb compounds that have been developed in recent years, there's often this talk of them being, not drugs of choice relative to, say, psilocybin and LSD. So it seems almost like you need some what do they call it, polypharmacology with the more well-known familiar psychedelics relative to the more selective ones. And there it's the effects peripherally are quite unpleasant, and 2A in the periphery and likely in the brain is vasoconstrictive, and that can be dangerous. So that's an issue, and people have overdosed with some of these novel psychedelics that are very potent, and they've mistook them for other compounds and taken too high doses, and some of the vasoconstriction has been part of the complications there.
The next question is from Richard Brockman, who asked if you could comment on the potential relationship between your rebus model and hippocampal-mediated reconsolidation of memory.
Yeah. I mean, it's a curious, the hippocampal reconsolidation. there's some interesting work around, I think there's this really neat title, something like a little prefrontal cortex goes a long way or something like that. So, I think of this relationship, well understood and characterized relationship between PFC to hippocampal circuitry, where the frontal cortex has a suppressive inhibitory influence on the hippocampus. And if these are compounds that are working principally in the cortex and principally high up, and particularly in other animals where the frontal cortex is more obviously hierarchically high level, less so in humans because high level is more distributed. It's frontoparietal and temporal. Then perhaps you're taking the brake, the top down from the frontal cortex, you're taking the brake off hippocampal function so that you can more of a sponge and you can learn more easily. That's one model. I suppose that would fit with rebus because you're taking the weight off the top down and allowing for more learning. Very simplistic, but that would fit.
So we only have a couple minutes left and there are a few more questions. So I'm just gonna give them to you sort of rapid fire. So someone asks about sex effects, difference in male versus female subjects in terms of their responses to psychedelics.
That's an interesting one. Nothing's come through, but that's a reminder. That's a student project. We've got a lot of data there and we can drill down into it. I mean, it's a little bit like a hemispheric thing. It's easy to find false positives. Of course, you've got, 1 degree of freedom there. It's left and right, male or female. Well, and we've found some things in our naturalistic data that went one way, and then in other data with other outcomes going the other. Basically, I can't say something super clear on that yet, other than it's a reminder to drill down into it. And of course, cycling in females is likely to be relevant and hasn't been well enough looked into. We do collect that data. That's another student project. Yes, so absolutely, we need to look into that one. Great.
So I think the last question gets back to this concern about the potential for addiction, but asks about increased neuroplasticity and potentially increased BDNF, and whether there's a potential, or should we be concerned about sensitizing various brain regions, particularly reward-sensitive brain regions, to future drugs of abuse, such as cocaine?
Well, that's a fascinating one. I mean, there's no evidence in any of the population or naturalistic data of increased addictive behaviors after psychedelic use. The data's in the other direction. We've got data on things like improvements in healthy lifestyle, eating, exercise, less smoking, less drinking after psychedelics. So, but I don't want to. I want to pause there to say that conceptually, yes, that if you're increasing plasticity acutely, and imagine that you're doing that in a more protracted way with microdosing, then you're going to be more sensitive to what you're exposed to. And it is an interesting ethical question of what are the implications of that, beyond mental health. We've looked at some interesting things around beliefs, philosophical beliefs, metaphysical beliefs in the nature of reality and self, mind-body relations and such like, and political beliefs and ecological beliefs and relationships and nature connectedness. And we've seen changes in an interesting direction, towards libertarianism, towards more nature connectedness. But is that because the dominant cultural context for young people, and particularly young people interested in psychedelics, is of that, of that direction. Also shifts in metaphysical beliefs, away from materialism and physicalism, if you want, and towards more of a dualistic perspective. And so there is this ethical question of, if this holds up, and it almost seems, is it context independent? Is it an intrinsic action of the drug that it moves people towards libertarianism or anti-authoritarianism? It's so hard to say. Yeah, but if that's a reliable thing, then it needs to be part of informed consent. It needs to be part of the narrative, and we need to think about it. I'd stop short of making any value judgment about those changes, but if they're real and reliable, then we need to know about them. Yeah. I mean, this fascinating phenomenon of the QAnon shaman, did get me thinking, this sort of alt-right, populist sort of political perspective, and an interest in psychedelics, and just got me thinking, plasticity times a certain information rabbit hole, takes you in a particular direction. Yeah.
Wonderful. So we are about out of time, and so wanted to really thank you for presenting to us, and I hope you could tell from all the questions that were coming in that there's a tremendous interest in your work, and it was just terrific to hear about all the different areas that you've considered, from high-level conceptual models to molecular pharmacology to clinical trial to QAnon shaman. So thank you, and really appreciate your talk. And if you are able to send your slides, that would be wonderful.
I'll do that.
Thank you again.
Thank you.